Hi everyone, good afternoon. So this is going to be the amino acid degradation section from the professor's notes in Biochem. I just took out the portions that she highlighted, and now this is what I have written down for it. Okay, so amino acid degradation, but first she wanted to give us like the summary of catabolism, amino acid catabolism. Okay, so first part would be arginine and glutamine. These are the two that are one steps, so we're only supposed to focus at the one step ones. So arginine and glutamine form glutamate. Alpha ketoglutarate will then come from glutamate, and that's going to form succinyl-CoA. The ones that make succinyl-CoA are isoleucine, methionine, threonine, and valine. And then succinyl-CoA becomes fumarate, and then those are tyrosine and phenylalanine. All amino acids are glycogenic except these two. So now we're talking about degradation. So lysine and leucine are the ones that are the exception to all amino acids are glucogenic. We also have ketogenic amino acids, which are usually the aromatic ones as well as the branched ones. The ketones will be in the liver. Right, catabolism cofactors. So in catabolism, we have biotin, which is going to carry CO2. We also have the tetrahydrofolate. This is for the CHOH, or the C double bond to O, or the CO2. So those are going to be those oxidative intermediates. Next, we have the S-adenosyl methionine, which carries methyl groups. Okay, so pyruvate amino acids, they're again going to be 3 carbons long because they're pyruvate amino acids, and the two that are the main focus are alanine and serine. Alanine requires an alanine aminotransferase, and again, aminotransferases, you need PLP for their reaction. And so alanine is going to become pyruvate. You also have this alpha ketoglutarate becoming glutamate at the same time whenever you have this aminotransferase. Okay, so now looking at serine. Serine will use a serine dehydratase, and that also requires PLP, and you're going to make pyruvate. The alpha ketoglutarate amino acids, these are going to be 5 carbon molecules. We've seen this before, so glutamine will require glutaminase, and that will make glutamate. Then you take glutamate and use a glutamate dehydrogenase, and that's going to form alpha ketoglutarate. Also, in the same respect, isoleucine, leucine, and valine are going to degrade in the muscle. These are the branch chain alpha keto acid dehydrogenase complex amino acids. So that's just another category. Okay, so oxaloacetate amino acids, these are going to be 4 carbon molecules. You have asparagine, and then you're going to be using this enzyme called asparaginase. That's going to make aspartate. Now, with your aspartate, you're going to use aspartate aminotransferase. So again, aminotransferase, we need a PLP. We need that pyridoxal phosphate. That's going to make oxaloacetate. And then, of course, combined with the aminotransferase, again, we have alpha ketoglutarate and becoming glutamate at the same time. Okay, so now amino acid synthesis. So nitrogen fixation is when we take NH2 to NH3. Aminotransferase, the PLP of the aminotransferase, will transfer that amino group from the amino acid, and it's going to form this alpha keto acid. So remember, there's a relationship between the alpha ketoglutarate and then the alpha keto acid. The alpha keto acid is the amino acid without that amino group on it. All right, cofactors for anabolism this time. So we have PLP, aminotransferase for the aminotransferase, and then we also have the same ones from catabolism. We have the tetrahydrofolate for those oxidized intermediates containing carbon, and then we also have the S-adenosyl methionine, which carries the CH3. And let me find where okay, there we go. So first, alpha ketoglutarate will become glutamate, and then glutamine. Remember, we've seen this reaction before. So you're going to again add NH4 to the amino acids. You're going to do several transfers of that. This enzyme here that takes alpha-ketoglutarate to glutamate is just an aminotransferase, so you need PLP. To go from glutamate to glutamine, you'll need a glutamine synthetase, if I believe. It's a synthetase you need ATP for. Let me see. Let me go back here. Yep, it's a synthetase. So glutamate to glutamine, you need a synthetase for that, so you will need ATP. The next one is 3-phosphoglycerate, and that will become serine. The third one is phosphoenolpyruvate, will become tryptophan, tyrosine, and phenylalanine. Number 4 is pyruvate to alanine, using that alanine aminotransferase. This one's a one-step. Number 5 is oxaloacetate, and then you have that going into aspartate and then asparagine. Remember, we use asparaginase to go from aspartate to asparagine, and then we also use the aspartate aminotransferase to go from oxaloacetate to aspartate. So plants and I think bacteria are able to synthesize all 20 amino acids. However, humans need half. We need to supply half of these amino acids. The ones that we can't make ourselves are known as essential amino acids. For example, there are all these like non-essential amino acids, and those are the one-step processes that we've gone over. And an example of a semi-essential amino acid would be tyrosine, since it's only made when phenylalanine, which is an essential amino acid that we can't make. Only when phenylalanine is present can we make tyrosine. So how do we make tyrosine from phenylalanine? So we're going to take this phenylalanine hydroxylase, and that's going to add an OH to the structure of phenylalanine, and that's going to get to our tyrosine. Genetic defects in this phenylalanine hydroxylase or any of these other enzymes will lead to inheritable diseases. All right, that's going to be everything for that chapter. Please do something nice or something.